In 7.2.5, they want to talk about how RNA and let's just say messenger RNA needs to remove something called introns to make our final copy of messenger RNA. So let's just change the color here. So if you remember, when we're talking about transcription, we have our nucleus with our genetic code in it, right? The DNA is not able to leave. And outside here, we have some ribosomes, which is where the proteins are made. So we need to get the message out. And we talked about how all we need to do is transcribe through the process of transcription is make a piece of messenger RNA, and then that messenger RNA can go out to the ribosome, and eventually that will make the protein that we want to make, which is called translation, and we will talk about that later. So that's what we talked about. However, our body is pretty tricky because what we can do is we can get many proteins from the same segment of DNA. So remember, a segment of DNA that codes for a gene, that codes for a specific protein is called a gene. So if you can think of this one blue area of our DNA as a gene, it makes this specific messenger RNA. But the trick is that it can change this messenger RNA after it's been transcribed. After we've made it, we can modify it. And what it does is it takes out parts of it. It's like, okay, we don't need you, and we don't need you, and now we've made like this shorter piece of messenger RNA. That is a big difference, right? Because this message, if it's smaller, is a different message than this one. And that's what this is saying. We can take pieces out and only leave the pieces that we need. So the pieces that we cut out and stay in the nucleus, they are not coded to make a specific protein. Those are called introns. And the way that I remember this is that they stay inside the nucleus and they don't leave. Exons, however, are the parts that get glued together and will eventually be this mature piece of messenger RNA that will code for our eventual protein. Let's talk quickly about how that works. There's a couple different steps here to explain like how this looks. More details here than you probably need to know, but it's always good to know some stuff. So in this diagram here, we can see that we have one intron in the middle. And if you remember, the intron will be the part that leaves. And then we have our exons over here. So the exons are the part that we want to save. So there's these molecules. It's like a combination of RNA and proteins that come over and they kind of identify the area that we're going to cut out like a scissor. These combinations of RNA molecules and proteins are called spliceosomes. So they're going to cut out this intron and it will be recycled, right? This will be recycled back inside the nucleus. And then we have this mature piece of DNA. This mature piece of messenger RNA. And that will leave the nucleus, find a ribosome, and eventually code for a specific protein. The one thing that I don't have here, that's another little modification that we make, is there's something called poly A caps. So what we do is that at the end, like before it leaves the nucleus, we put adenine RNA molecules, RNA nucleotides. We put adenine on both ends to protect it, right? Because as this thing's like traveling around in the hazardous environment that is our nucleus, we don't want to bang up any of the important code. So if we lose one of these, no big deal. If we lose one of these, no big deal. But these poly A caps go on here to protect it. So we've got this mature piece of messenger RNA, but maybe the question is like, why? Like, why do we need to have so many different types of messenger RNA from one gene? And the best example of that is our immune system. So our immune system, the protein that it makes, that it might be most famous for, are antibodies. So let's pretend that this is the genetic code for one particular antibody. And we have these different colors here that represents our exons, the part that will be the mature RNA, and these parts here in between, what are they? The ones that stay in the nucleus, they are introns. So let's say that our messenger RNA, one of the options is that we use all 5 exons and we glue them together like this. So remember that this messenger RNA will eventually make our protein. Here we have a diagram, maybe with an alpha helix here, another alpha helix here. Maybe we have some, if you remember our amino acids that are hydrophilic might be on the outside. Maybe there's a cysteine here that's causing it to come together, right? So you have all these different characteristics of our protein that create a specific shape. So this antibody, if we're talking about antibodies that are going to help us, that's what IgM means. These antibodies are going to fight a disease. You know what? Diseases mutate. We have a different cold and a different flu every year. So it's important for our genes to also adapt. And I'm not sure of the mechanism, but it's able to reconfigure our original code. So in this one, in number 2, it's like, you know what? Different disease. I need a different antibody produced, so we're not going to use the genetic code on exon number 3. So this mature messenger RNA is only going to be composed of exon 1, exon 2, exon 4, and exon 5, which means we have a totally different looking, right, polypeptide. All of this has been removed, and now we have a different shaped protein, which means it will have a different function. And that function would be providing us immune protection when we're infected by a disease, which we will talk about in 6.3, and it's super interesting. So that is the deal with introns and exons. Introns are not used. They stay in the nucleus. Exons are used. They are the code that enters, that will eventually leave our nucleus and form that mature piece of messenger RNA that's used to express a specific protein.